Biology chapter 19 theory video 9 gel electrophoresis. uh, Gel electrophoresis is actually a technique that can be used to separate different molecules, and also it can be used to separate DNA by separating fragments of DNA. To analyze DNA, we can use this technique. So what happens is these DNA samples would be placed in small wells cut into a gel, and onto that we can apply an electric field, because the negatively charged DNA, the DNA is negatively charged because of the phosphate part in it. So it will actually get attracted, or it would move towards the anode, and the positively charged molecules will move towards the cathode. Then highly charged molecules will move faster than the less charged molecules. Then the size will also matter. The smaller molecules will move faster through the gel in comparison to larger molecules. Then the composition of the gel will also matter. The size of the pores will also have an effect on how fast it would move. So here DNA will, like I said, it has a charge because of the phosphate group on it, on the backbone, or the sugar phosphate backbone. So they move towards the anode, and then how much it travels will define the size of it. So the distance traveled by the length of the DNA is going to be inversely proportional to the length of the DNA molecule. The shorter it is, then the farther it is going to move through the gel. So the procedure is this, and also there are two types of gels that can be used. Polyacrylamide gel is used for small fragments of DNA that may differ by as little as 1 nucleotide in length. So agarose gel is used if they are separated by fragments between 100 and 50,000 pairs in length. So the procedure is, you take this gel powder, mix it with hot water, and while it is still a fluid, you pour it into a electrophoresis tank. There is a comb placed at one end to make the wells or kind of holes in it. And when the gel is solidified, the comb is removed, and the samples are loaded onto this. So when the gel is set, there would be a buffer solution poured onto the tank, so it should cover the gel. So this gives a constant pH. Then a micropipette is used to transfer the samples of DNA to all of these wells, and the samples of DNA will have a tracking dye which can show how far these materials would travel across the gel. So it is quite similar to that of the chromatography solvent. It has the almost similar function. Then most often we apply reference samples of DNA fragments with known lengths to at one side or either side of on both side of the gel. So that will create a DNA ladder. So this DNA ladder will be used to determine the length of the fragments of the DNA that we are going to check. Then a battery pack is connected to the electrodes. Then the negative electrode is at the same end as the well. So the negative end is at that point. So as a result, we can see the positive side is on the other side. So as a result, negatively charged DNA would move to the other side. So when the dye, the tracking dye will show how far it has moved. When the dye has moved through most of the gel, we have to disconnect the battery and start taking measurements. So after the battery has been disconnected, we pour out the buffer solution, and using a stain we can figure out we can actually see the positions of the DNA fragments, and we can also use probes using fluorescent stain. So somehow a stain is used to, that is all you need to know. A stain is used to find the length of the DNA fragments by comparing them with the ladder at the side of the gel. DNA in the nuclei of eukaryotes has short nucleotide sequences that are repeated. They are repeated quite usually many times over. Now, for example, if you take a pair of homologous chromosomes, the base sequence ACA, they might be repeated 65 times on one. On the other, it might be repeated 118 times at the same location. In both of them. So they have many variants with different number of repeats of these base sequence. So these base sequences are also inherited just like the way alleles of genes are inherited, and these repeated sequences actually gives us a genetic fingerprinting to identify different people differently. So these are actually initially they used gel electrophoresis. You use the if the number of repeats are greater, the fragment of DNA is going to be longer. They used mini satellites. So the first repeated sequences were mini satellites, and later on they started using microsatellites. So they are much more shorter. uh, 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 these mini satellites are known as variable number tandem repeats. They are used in the first form of genetic fingerprinting to give the ladder-like bands on gels. Then short tandem repeats, they are microsatellites. They are repeated sequence of nucleotides which are much shorter than mini satellites. So they are composed of 2 to 5 nucleotides which are repeated 10 to 30 times. So these are actually just some parts that you have to remember as theory. Don't try to go into details of these things. When you look at the past paper questions, you will understand that they are not going to ask you the process, but rather the general idea of these usages, right? So they actually developed for example, FBI, Federal Bureau of Investigation in the United States. They develop these databases using these the details that they obtain through uh, these analysis to identify different people. Write down a note on this. Gel electrophoresis. This is a technique by which charged molecules are separated by differential movement through a gel in an electric field. The degree of movement is dependent on the mass of the fragments of DNA. Following factors will determine how far the charged molecule would move through the gel. Number one, overall charge. Highly charged molecules move faster. Smaller molecules move faster, and the size of the wells or pores within the gel will determine the speed at which the DNA move. There are two types of gels being used: polyacrylamide gel and agarose gel. Polyacrylamide gel is used to separate small fragments of DNA that differ by as little as one nucleotide in length. Agarose gel is used to separate fragments that are between 100 base pairs to 50,000 base pairs in length. Leave a line and write. Procedure. Gel is made by mixing the powder with hot water. It is poured into an electrophoresis tank using a comb. Wells where samples could be loaded are made. After gel is set, a buffer solution is poured to maintain pH. Using a micropipette, DNA samples are filled into the wells, and the samples would have tracking dye which would travel along with the DNA. Reference sample of DNA will also be added to one side or both sides of the gel, and this will be used to determine the lengths of the DNA fragments in the sample. A battery pack provides the electric field. After the dye has moved a certain distance, buffer solution is poured away, and using an appropriate stain, the DNA ladder would be observed, which can be used to analyze the size of the molecules. DNA in eukaryotic organisms have short nucleotide sequences that are repeated many times. They have different number of repeats at different locations. These repeated sequences are unique and are called genetic fingerprinting. Initially, mini satellites having base sequence of between 10 to 100 base pairs were used. However, now microsatellites of between 2 to 5 nucleotides are used. Microsatellites are known as variable non-tandem repeats, VNTRs. Microsatellites are known as short tandem repeats, STRs. These mini satellites and microsatellites are found on the 22nd autosomal chromosomes as well as both X and Y sex chromosomes. In in genetic profiling, the STRs in the regions of non-coding DNA between the structural genes are used. And I hope you understood. If you have any questions, you can ask. Please go to question number 11.